So I'd like to introduce Ben Mayne. Ben is a molecular biologist and bioinformatician with the CSIRO in Perth. Ben's expertise is in analysing changes in DNA, known as epigenetic modifications. These DNA modifications can be used to determine important but often difficult to determine characteristics for wildlife managers. Ben is currently focusing on a project to predict the age of animals using their DNA. The aim is to develop a non-lethal and rapid technology to predict the age of animals to improve wildlife management. In this talk, Ben will show how to predict the age of freshwater native fish and marine turtles using their DNA. Join me in welcoming Ben.
Well, hi everyone. Thanks for the organisers for giving me the chance to talk. It's nice to actually do one in the flesh as opposed to like online for the last year and a half, so it's a nice change. But as Chris was saying, pretty much I'm just going to talk about the project that I work on, which is trying to predict the age of animals using their DNA. The reason why we're so interested in age is that we're trying to generate these sorts of graphs, which you may have seen when it comes to human populations. So you've all would have filled out the census recently, and one of the questions would have been, what is the age of all the occupants within the dwelling? And population demographers use that sort of information to work out whether or not they should build a new primary school, or should it be more retirement homes and all that sort of stuff. But with animals, age is really important to work out a number of things. It can be used to work out the risk of animal extinction. For invasive species, it can be used to work out if there's an upcoming population boom. But mostly in fisheries, age is used to work out how many can be the sustainable harvest each year. Unfortunately though, most animals don't really have a practical or non-lethal method to work out their age. So at the moment, the traditional methods is looking at growth rings in bones to work out the age of animals. So in fish, what occurs now is that they chop off the head and then they extract the otolith or the inner ear bone, and there's basically growth rings which get put down annually, and that's pretty much and so each ring is basically a year, sort of like how you would count rings in a tree to work out age. A similar process can also be carried out in marine turtles, where basically you can make a cross-section of the humerus bone. However, for a conservation setting, it would be counterproductive to kill animals just to work out their age, so we've been trying to develop a more non-lethal approach. So on the molecular level, or on the DNA, there's a process called DNA methylation. In this picture here, it's the CH3s in DNA. Basically, this is a process which is probably more known for regulating gene expression. So certain tissues will only express different genes, and this is the primary method of how cells control that. However, it's been shown that this process is also known to change with age. So as cells divide, cells are meant to maintain these changes during cell division. However, cells can be a bit sloppy, and so they're accidentally putting down methylation marks in the DNA where there should be, or taking away marks where there otherwise should be. And so basically what we're measuring is cell senescence. This method has been used to work out the age of predominantly mammals so far. So it was first done in humans. It's also been carried out in whales, mice and dogs. These studies were really... wrong mouse, yeah. These studies were really great in that they produced really accurate models. For humans, you can get it down to within a couple of months, depending on the sort of test. However, unfortunately, they carried a very high cost of about $500 an animal. And when you're sort of trying to manage, get these sort of population studies for wild animals, you're going to have to start sampling hundreds of animals. And for that $500, that's just simply too expensive. So the way we do it, so what we've been doing is sort of taking that technology that's been predominantly done in humans and applying it to wild animals, mostly fish and turtles at this stage. So what we do is we take a small tissue sample from a fish or a turtle. This could just be a skin biopsy, some saliva or some blood. And basically we just measure these changes in DNA methylation. This is, we're basically looking at sites in DNA that we have already examined to know the change in DNA, change with age. And basically we developed these sort of algorithms for different species to work out their age. So that's sort of like the pipeline that we use. And just in terms of the cost, what we've just done recently is developed a more cost-effective strategy. So what those those studies that were doing it in mammals and humans, is that they were looking at all the changes across the genome or the entire string of DNA. Whereas what we've found is that you only really need like a subset of sites in DNA to look at. And so in terms of when you sort of sequence this DNA, it really reduces the cost. So we've gone from $500 to $5 per sample now. And this makes it quite effective, or a cost-effective method to use in a wildlife management setting. So we've done it on quite a few species so far. This is just a subset. So we've been doing it on mostly native freshwater fish. I'll talk about the Australian lungfish in just a moment. But we've also been doing it on marine animals as well. I'll talk about the turtles as well, but we've also done it on commercially fish species such as tuna, and we've just starting to do it in things like school sharks as well. And the idea is that we'll, at CSIRO, that we'll be doing this as an annual thing, and this will provide wildlife managers for a more precise population, an overall population of how many animals there are in the wild. So just to give you two examples, so the first is with a lungfish. For those who don't know, there's only 6 lungfish species in the world. Only one of them occurs in Australia. They're found in southeast Queensland in only 3 rivers. So there's the Burnett, Mary and Brisbane River. The population is just really understudied at the moment because they're known to be a long-lived species, but no one knows how the ages of them, and that sort of makes it difficult to work out for population growth because they're only reproductive at certain ages. Also, the rivers are also undergoing a lot of dams at the moment, and there's a lot of recreational fishing. So although these particular fish aren't eaten, just the sort of traffic within the waterways does sort of make it a bit difficult. Makes it basically it puts the population under threat. So there's a real need to work out, get a population assessment. So what we've done is we've taken lungfish of known ages, so these are ones from mostly aquariums across the world that have been shipped off from many years ago, which I'll talk about in the next slide. And then we've looked at the changes in DNA to work out, to develop a model to work out that age. So in this graph here, on the X or the horizontal axis is the known age of these lungfish, and on the Y axis is the predicted age from the DNA. And as you can see, and each dot is an individual fish, and as you can see, it produces this very nice straight line, which suggests that the model is predicting the ages very well. And in fact, we can get it down to less than a year in accuracy. Just to give you one example, so a lot of those fish of known ages were ones from captivity. So what happened during the 1930s, a lot of scientists from Europe and America came to Australia, and they were really interested in lungfish because it was sort of that sort of missing link in terms of evolution. It's considered a living fossil. It has has this really only distinct evolutionary line. And what happened in the 1930s is that these were all just sent around the world, and one of them, which is referred to as Grandad the lungfish, ended up in Chicago in time for the 1933 Chicago World Fair for whatever reason. But this gave us a great opportunity to actually look into the longevity of the species as well. So Grandad ended up living at the Chicago Aquarium up until 2017 when it was euthanized. But since it was already an adult when it was taken from the wild, they never knew how old it really lived to. However, using the model that we just produced, we worked it out to be 108 when it died. That has sort of implications of how it's also managed in the wild, so knowing that it can go past 100 years and that they might actually have a late maturity when it comes to sexual reproduction sort of gives you an idea of how to how population growth might be into the future. And just as a side, the records were sort of a bit blurry, so they didn't know which river it came from, and however we were able to look at the genetics and compare it to the known genetics of the different river populations, and we were able to work out which specific river it came from, and which in Bunbury at least they were very excited to find out about it. So something a bit more Western Australian focused, we've also been developing a model for green sea turtles. Again, this graph here was taken with turtles from captivity. Most of them came from the Cayman Islands. Just for those who don't know, in the Cayman Islands there was an attempt to actually farm turtles. Apparently they taste very good. However, it wasn't a viable business, so there's just a wildlife sanctuary now, but there's a whole bunch of turtles that are up 40, almost 50 years old that were intended to be eaten, but fortunately we were able to take the tissue from these turtles and actually develop a model. So as you can see here, we get this straight line again, and in terms of accuracy, we're going to get to about 2.5 years. Not as accurate as the Lungfish model. That's mainly because we don't have as many samples. And that's really the limitation with all this sort of work. We have to compare it to known ages to begin with. So when it comes to other turtles, like the leatherback and flatback sea turtles, this makes it difficult to generate these sorts of models because we don't have access to known ages. So to get around this, what we're trying to do is use the lifespan of species to recalibrate models where we have already generated for some species and then transfer it into others. So it'll make sense in a moment. But in short, what we've done also is, just to go on a slight tangent, is generate a model to work out the lifespan of species. So a lot of long-lived species might outlive a generation of researchers. And so it might be very difficult to actually get a lifespan estimate. And so what we've done is we've looked at the genetic sequence of different species, and then we've been able to work out, by looking at these sequences, to work out the lifespan of different animals. And we've generated a model which is a bit noisy, but can work out the lifespan of different species. So we've applied this to a whole bunch of different animals, including ones that have gone extinct. So using the DNA of a mammoth, for example, we've been able to work out it was most likely lived up to 60 years old, which is similar to what an elephant does in the wild today. But using this sort of information, we've been able to work out the lifespan of different marine turtles. So these are all the different marine turtles that occur in Australian waters. So the most long-lived one is the leatherback sea turtle at 90 years, whereas the flatback sea turtle is the most short-lived at 50 years. And now, so although these turtles might not necessarily live up to these ages in the wild because they might be under threat from various anthropological sources, but since we now know the ratios between these lifespans, we can then apply it to our green sea turtle aging method to work out the ages of different turtles. So the idea is to take that green sea turtle model that I showed you in the last couple of slides, and then using the molecular lifespans, the idea is to then to recalibrate it for these other turtles where we don't have access to known ages, if that makes sense. Sort of like a long way round to get to the answer that you want. But just to give you an idea of what we're sort of using this information for, in Ningaloo at the moment, we're sort of trying to generate trying to do a population assessment at the moment. Unfortunately, just trying to get the turtles is quite difficult to begin with, just to get a tissue sample. So what we're doing is, firstly, we're taking about 300 turtles at the moment, where we've measured their lengths and then compared their lengths to their ages, and we found a relationship between age and length. And then on top of that, what we're doing with other collaborators is flying drones across the reef, and then this is able to find different turtles. And then from these images, we've been able to work out the lengths of these turtles, and then since we know there's a relationship between age and length, we can, from these photos, get an approximate estimate of their ages. And this will save actually having to handle turtles into the future, and then just by simply flying a drone, you might be able to get a reasonable assessment of totals within a given area. So that's pretty much it, but the overall idea is that we're sort of generating these what's referred to as epigenetic clocks or DNA-based clocks to work out the ages of different animals, and we're hoping that this sort of information will be able to improve wildlife management in the future. And with that, I would just like to say thank you again to giving me the opportunity to talk, and everyone who's been involved in the project so far. Thanks.